and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 163 for the podcast. Lonza is one of the world's largest contract development and manufacturing organizations dedicated to serving the healthcare industry. They work alongside a broad range of customers, from emerging biotechs to top global pharmaceutical companies, to transform therapeutic discoveries into life-saving and life-enhancing treatments for patients. Founded in 1897 in the Swiss Alps, Lonza has embraced innovation and kept pace with a fast-changing world for more than 125 years. They are a leader in both established and emerging technologies, and today their 19,000 employees across more than 30 sites on 5 continents generate annual sales of more than $4.4 billion. This week, I sat down with Alice Harrison, Global Technical Director, CMC and Analytics, and Megan Mason, Global Process Development Implementation Manager, to learn more about how Lonza is helping innovators large and small develop and scale their therapies and deliver better outcomes for patients worldwide. I hope you enjoy our discussion. Megan Mason and Alice Harrison, welcome to Beyond Biotech.
Thank you.
Megan, I'm going to start with you. Can you tell us a little bit about your journey in the industry? What led you to your role now, and what exactly do you do there at Lonza?
Sure. So my background is very much in chemical engineering. So I actually started off in the US at Rensselaer Polytechnic Institute. And once I graduated from my bachelor's degree, I started off in small molecules doing cleaning validation studies, which wasn't very glamorous, and quickly realized that to do what I needed or wanted and aspired to do, I needed to go back and do an advanced degree. So I went back and started on my PhD, which took me to the UK and introduced me to the Lonza GS expression system, as I was partnered with a company who was looking at how amino acid changes impact expression of monoclonal antibodies. That very much introduced me to Lonza. Once I finished my PhD, I ended up joining the cell culture development team over at Lonza as a bench scientist, and worked my way up through the team. And it brings me to where I am now. So I am in the global process development team, and my job is an implementation manager. So I'm looking at the rollout and alignment of technologies across the Lonza network to make sure we're doing things in a consistent way, so that our customers get the same experience no matter where they choose to utilize our services.
Perfect. And Alice, yourself, how did you find your way to Lonza?
So I'm a bit more of a recent recruit to Lonza. So I've been in Lonza about 3 years now, but my journey started quite a long time ago. I did a first degree in chemistry, and my second degree I went into molecular immunology at Oxford. And during that time I was working on T cells, and in particular molecules that trigger T cells and can cause things like autoimmunity or unwanted immune effects. And during that time, it was really the early days of biological molecules. And I was working on a molecule that was one of the very first molecules that ever became a biologic. So that was CD58, which binds to CD2 on T cells. And this was really exciting to me because it was being developed for an autoimmune disease of which I was a sufferer at the time. And it was so exciting at the time to believe that this was a new wave of medicines and therapies. And I was one of the first people, I think, in the UK to have another biologic called etanercept, and then followed on with antibody biologics. And it's not a cure, but it certainly meant that I could live a completely normal life. So really from that moment on, I knew that this was the industry I wanted to stay in and help to get medicines to market. So over the last 25 years or so since then, I've developed my journey across the CRO, CDMO space, helping companies to take their biological molecules to clinic. So when I started my role at Lonza 3 years ago, it was really to support companies developing these molecules. So I'm a technical director in Lonza, and I support the proposal process, the operations teams, in order to get these molecules into the clinic and ultimately onto the market.
Alice, it sounds like your personal history there really helped you to focus on bioprocessing and that innovation. Did it really shape your perspective?
Absolutely. I genuinely felt I wanted to be one of the scientists who helped people in the way that the scientists before me had helped me and other people. And my disease is not oncology disease, so I'm not going to die from it, but it was very debilitating. And being able to see these innovations and work on them in the laboratory at the University of Oxford really stepped my path forward. And the innovative approaches have continued from then onwards over the last sort of 25 years. The molecules that we see and the things that have been developed have really surprised me every day. So the new tools, the AIs coming through, all of those innovations are just incredible, and it definitely inspires me to do what I do every day.
And Megan, you came from academia, you did your PhD, and then you came to such an innovative company. Is there something in your background that made you just decide that Lonza was the place where you wanted to be?
So I did, before I went down, or in parallel to deciding what kind of my future career was going to be, I did toy with going into medicine and to become a doctor, and then decided that maybe patient interactions weren't for me. So I knew I wanted to do something in pharmaceuticals, so pharmaceutical manufacturing seemed to be a nice fit. And one of the things that drew me to Lonza is that we work with a lot of different companies, from very small startup biotechs to really big players in the pharma industry. And once I joined Lonza and started working with customers, one of the nice things is we get a lot of customers coming in and telling us about their patient stories. So often getting patients to give a little snippet of what we're manufacturing drugs to treat. And in that, especially the ones that have like very niche unmet need, you see like how important getting these drugs out to market fast is. And so a lot of the work that we were doing when I was in cell culture development was all about really decreasing those timelines. So I could see why there was a need to innovate and go faster and drive the costs down. So not only were we kind of getting those drugs out there as quick as we could, but also, trying to keep the cost reasonable so that, health authorities would be willing to license them, especially with those really kind of niche patient sets. So, it's been an interesting journey, and one that I think will continue to be challenging and continue to need innovation, right, to keep pushing those limits.
Why don't you tell me a little bit about Lonza, the company, Megan? They've got a real history as a global leader in bioprocessing. Can you maybe give me an overview of what their mission is, though, and how it supports the industry who are trying to bring these therapies to the clinic and eventually to the market?
Absolutely. So, I mean, Lonza's vision is to be the world leader in the CDMO space, right? So, we hope to achieve this through constant innovation in our technologies and our platforms, but is combine these alongside kind of lean manufacturing practices, right? And so to do this, we really aim to work with our customers. And like I said before, we work with everyone from very tiny kind of virtual companies to the massive players. And their needs are all very different, and each of the molecules' needs are very different. So especially as we see we're shifting away from kind of the traditional monoclonal antibodies into more complex molecules, we really do need to sit down and kind of create work packages that work for our customers in order to deliver what their goals are. And so we've recently restructured the business into 3 different business platforms. So we've got our integrated biologics platform, our advanced synthesis platform, and specialized modalities. And what we're aiming to do is kind of work synergistically across these 3 to kind of deliver those requirements of our customers. So yeah, that's what we're working. So we call it the One Lonza strategy.
Alice, maybe I can ask you here. How are you actually differentiating yourself on this CDMO landscape? Because just as there are huge pharma companies who would be clients of Lonza, there are also some pretty small biotechs there, and you're serving both ends of that market.
Yeah, that's a great question. I think what really sets Lonza apart in the CDMO landscape is we have a really unique combination of scale, scientific depth and history, and flexibility. So I feel that we're really uniquely positioned to support both the smallest biotechs. And we have virtual companies, so companies with absolutely no labs at all and very minimal staff, and large pharma, so some of the largest pharmaceutical companies in the world. And so we've managed to take a very flexible approach. So we never assume that one size will fit any company. So even two small biotechs that seem very similar can need a different approach. So we really tailor how we work based on each customer's needs and their molecule needs and where they are on the development journey. So obviously small biotechs are likely to be at the start of their journey. They're quite often spun out of academic labs, so they have a real need for speed. They need guidance. They might have no regulatory expertise at all, and so building that partnership and building that trust is really critical early on. Also, we find with small biotechs, they're often the ones working with the most innovative products, so it's quite a combination of lack of experience of these kind of manufacturing challenges, combined with the most unique, most innovative products, and sometimes the most challenging. And they often don't have the kind of large internal teams that they may require. So we become not just a service provider, but they can leverage our experience of regulatory experience. We've recently celebrating taking 100 molecules onto the market, and also the scientific teams. So we have teams within Lonza that have been working on these kind of molecules ever since these kind of molecules started to be manufactured. So, and it really helps small companies to make smarter and more compliant decisions about the developability of their products, and also help them to define that manufacturing strategy, which can really make a huge difference later on. Large pharma needs are often quite different, so they can be much later in the pathway. They may have a much more rigid focus on scalability, looking very deeply into our quality systems. Obviously, we have a long history there, and leveraging our global network. So we have large scale manufacturing sites across the globe. And so we meet those expectations of the large pharma, which may have very different needs to what a small biotech company needs, although sometimes even with large companies, they're developing new products and innovative products. And so they often leverage our technical problem solving skills and our cross-functional collaborative approach.
Megan, Alice mentioned some innovations that are happening outside of Lonza. What about innovation within Lonza? What are the sorts of things you're currently advancing to support the next generation of biologics?
Yeah, so we're focusing on approaching innovation from several different areas. So kind of looking at kind of the earliest technologies in which we start working with a molecule. So around the vectorology, we've just launched the G Squad Pro system. So that's building upon the previous G Squad vector system, which allows up to 4 genes to be expressed in a single vector, which really helps with kind of the new molecular formats, so bispecifics and other kind of more complex proteins. We can look at playing around with the gene order within that vector system to be able to optimize the expression as well as make sure we're maintaining the correct product quality of that molecule. And G Squad Pro is now coupled with a synthetic promoter that we've just recently launched. And so that actually gives us even higher expression. So it's about 40% higher on average compared to the previous promoter that we were using. So we get more out of our production process, and also the cell lines tend to be more stable, so they're more suitable for really large scale manufacturing. So you can be confident basically that the molecule that you're expressing kind of in tiny little flasks is going to scale all the way up to our biggest tanks at 20,000, 25,000 liters. So we can also couple that kind of the vectorology elements with our piggyback transposon technology. So that's ensuring that the genes that we insert into the cells are going into areas of the genome that will be highly expressed, and again, it should maintain that stability as we're manufacturing well up into those large scales. So we got the vectorology. Then we've got some innovations around the process itself. So we're looking at more kind of intensified processes within Lonza. So we've got a process that we've launched where we intensify the inoculum steps, that we then have a high inoculation density process within the actual fed batch tank itself. And so that can give you a titer uplift of about 80% compared to a traditional fed batch process. So when we use these synergistically, we're really kind of pushing the titers that we can achieve up, which helps reduce the cost of goods and keep the costs down in general for our customers. In addition to that, so we're getting more product back out. We also can kind of look at how we put the different process steps together in an offering from gene to IND. So we've got rapid tox offerings that we've recently launched. So often when we look at what the shortest possible timeline or what becomes the limiting factors in product development, the time to do tox studies is quoted as being a limiting factor. So we can actually, what we've done is looked at using pools, for example, to generate tox material, and where we have a formulation that we can put a molecule into, we can do tox material generation in as little as 2.5 months. So we kind of shift that off critical path and allow that critical kind of timeline compression. So when we take these things all together, we are actually in a position now where we're happy to tell a customer that we can get them a 5 gram per liter cell line, and it will be a stable one. And we've recently launched a guarantee of this, as Alice mentioned before. We've recently celebrated the 100 molecules market. So as part of that, yeah, we're taking all these innovations and combining them together, and it's exciting times.
It certainly is. Alice, things are also getting a lot more complex. So there are more complex biologics, the biospecifics, ADCs. As these development pipelines are evolving, what does it actually mean for a company in the CDMO space like Lonza?
Yeah, I think you're right. We're seeing a lot more diverse range of molecules coming through into development, and hopefully a reasonable number of those will make it to the clinic. But it does mean that we need to change our approach, and that our customers are also changing their approaches. And I think what we're seeing really is there's a shift towards an earlier integration of CMC thinking. So customers are starting to think earlier on about how will their molecule perform, not just in the clinic, but how it perform during the development and manufacturing phases, and looking for risk mitigation. So for a CDMO like Lonza, it means with us, we do more than just manufacture products, and we're being brought in earlier to help with robust, scalable designs and development of these products. So for example, we had a customer who knew what the targets were, and they often had a platform-like approach themselves to how they developed their bispecific and what they format that they wanted to be, but they were willing to explore different formats that could give them better results during development and manufacturing. So we screened a wide range of formats for them, looking at the aggregation potential, fragmentation, things that may cause us real challenges during development. And then through that screen, they were able to choose a better format, so take a step away from their platform and look at a different format in order to take their molecule further. So that's, I would say, a bit of a change as to where the CDMOs are in terms of actually helping to design better formats for customers. We're also able to do a lot of in silico screening, which can help look at liabilities. And it means that our capabilities need to keep up with this innovation. We've, as Megan mentioned, we invest heavily in new technologies, In the sort of development space, we look at automation, advanced tools that we can use to better support these molecules. And I would say one of the most important things is building teams that have a deep experience in non-standard constructs and molecules. So people in the team who really understand the nuances of these interesting and diverse molecules that we see coming through. And so the support model for CDMOs really needs to be, can you combine the technical depth? Can you adapt to what the molecules need? Because these kinds of molecules will always give you some kind of unexpected challenge. So it's being able to be flexible around that, being able to have the tools in place so you can rapidly adapt your development strategy. And I think those will be the CDMOs that really help to bring these next generation of therapies to market.
Megan, Alice is talking a little bit about how CDMOs are adapting, but what about on the other side? What sort of hurdles are biotechs and biopharma companies navigating as they move forward on this journey to being first in human?
So companies still face persistent challenges that we've seen in the industry for years, such as high attrition rates with the molecules that they're trying to get into clinic. There's still a push, as I said before, to get those development timelines down as low as possible and doing it on as tight a budget as possible. So what we aim to do with customers is to get it right as early as possible to kind of de-risk these approaches and still meet their timeline and cost specifications for a given program. So that starts with developing the strategy alongside these companies. So in early development, right, you want to use all the tools that you can to assess the product in a model of your manufacturing process so that you make the right decisions and ensure that your molecule has the correct product characteristics that you want to see from your manufacturing process when you're at clinical scale and ultimately, hopefully, at commercial scale down the line. But when we talk about trying to do things quickly and avoid costly redevelopment, sometimes customers need kind of bespoke solutions. So one example, we've had customers that have come to us where they've got several molecular candidates and they don't know which one they want to go with, but they know that they've got a time commitment. They have an IND filing that they need to hit by a specific date. So we've actually developed kind of timelines for that customer where we start their cell line development for multiple molecules in parallel. And what we can actually do is leverage the synergies of running those projects at the same time. So you can avoid replicated efforts. You can get the number of molecules that you're screening to fit on equipment and use the experience that we have of running these programs to help funnel these development timelines through so that you're building in kind of key decision points during their cell line development journey that allows them to parallel track those molecules and get key information such as maybe a tox readout to then select one to move forward, but they're doing that as quickly as they need to meet the milestone that's important to them. So it's one approach we can take, but we've got experience tackling it lots of different ways depending on what the customer needs, so that we've always got a starting point and then we build the solution alongside the customer so that we are making this decision alongside them and not independently of them.
Alice, why is it so important to get those really early decisions right when it comes to development? I'm thinking here of the IND phase or really early clinical phases. Why is it so important to make the right call early on?
I think it's a really important question, and decisions in the lab can really set the foundation for everything that follows. So risks or liability that you miss early on can cause you significant problems later down the line in terms of manufacturing or scalability or the stability of the molecule, or even introduce regulatory risks in terms of what you may be able to achieve for that particular molecule in that particular process. So we focus a lot of effort on this in the early phase, or as early in development as we can, because we know how costly it can be to correct for those liabilities later, or to have to change a cell line, or to have to redevelop a purification process. Those are the things we would rather avoid. So we try and get the design and the lab experiments right as early as possible. So I can give you an example of that. So during a manufacturing screen we saw for a customer, it had a high risk deamidation site in the binding domain of the molecule, and we knew that if that risk became realized, it would have an implication for the molecule's function and potency. So these are things that we can build into the development program. So in that case, we looked at all of the areas along the program where that might become a risk. So we know during the purification process, we may have to use pH in order to do the elution from the columns, which may cause the deamidation risk to be realized. So in that case, we looked at the mitigation strategies across the purification process in order to ensure that deamidation would not occur for the process that we developed, and modified the process as we went along. So we tailored the purification in order to meet that molecule's needs, because we knew it was such a high risk liability. And obviously, because it was in the binding site, we couldn't change the sequence. We couldn't go back and change the sequence of that molecule, because it would intrinsically change the biological properties of the molecule and the molecule's function. So by understanding the risks of the molecules, then we can really help to de-risk the program. Where we can, if we see a risk and it's significant enough, we would advise customers to think about their molecule sequence or the molecule stability, but in many cases that just isn't possible. The changes that would be required would mean the molecule doesn't have the same clinical function. So these kind of assessments and these early assessments and the early lab work that we do is really crucial for making the right decisions for the products later down the line.
So getting those early decisions right is really important. But Megan, what's Lonza actually doing to ensure that the assembly is correct, quality, the function of the complex biologics during that really early development stage are setting companies up for that success?
So Lonza offer a range of early development services, and this includes things like the in silico manufacturability assessment, which Alice alluded to. So we can actually look at what sequence liabilities might be present in a molecule or a series of different molecular sequences that a customer may still be considering. And so if you can engineer those out before you've even started work, happy days, right? So we can look at that, and we can advise on how to remove aggregation potential or deamidation sites or anything that you don't want to find when you're doing your molecular characterization much further down the road. So that can be a good starting point. We also offer things like in vitro immunogenicity screening. So you're making sure the safety profile of your molecule is as you would expect and as optimal as it can be. And then we also can generate really early material off of pools kind of at this early stage for multiple sequences as well in parallel. And you can start looking at things like your formulations, right? So you can do really early formulatability screens, which can then contribute to shortening those timelines, right? So you've got a readout of what formulation your product is going to be the most stable in. And that then feeds into the subsequent stages that follow on in your development activities. Right. So all of those help contribute to minimizing any headaches that you may come across down the line, as well as kind of looking at having a good safety profile for your molecule. The other thing that we've kind of mentioned previously, but it's important, is that we're using vectors that are manufacturing ready. And then we've got a cell line in a production process, right, that has really good kind of regulatory pedigree. So everything is done with ultimately your regulatory filing in mind. So as we pass through kind of the critical path, everything is done in a way that will support that regulatory filing at the end of the road, right? And by generating kind of that representative material up front, it means you can start other kind of critical activities such as the assay development, or if you're making ADC, you might start doing your conjugation evaluation steps as early in the process as possible. So again, these are all kind of foundational elements to accelerating that journey to kind of IND and that first in human.
Alice, maybe you know something a little bit about the assays as well. You've had some experience there.
So early in the development phase, we try and use assays and methods that will take us through for the whole program. So the results that we're seeing early on, so things like assays for looking at aggregations, such as size exclusion, are comparable and applicable to the later stages. So for the platform assays that we would use, so that we are getting comparable results and methods throughout the whole process and program.
Megan, choosing that right expression system, that's a really foundational step for any biotech or pharma. Can you explain how Lonza's GS system helps to optimize that early stage development? And can you maybe explain why it's really suited for these new molecular formats?
So the GS expression system, as we said, it's been around for a long time. It's got a long proven track record across a very wide range of molecules. So we've alluded to the 100 marketed products, but 20% of these fall outside the classic monoclonal antibody formats. And we're seeing an increasing proportion of the molecules that come into the system. We actually have more new molecular formats than we do monoclonal antibodies. So it's about 60% of the molecules that we see would fall outside the classic monoclonal antibodies. And so we've talked about how the vectorology that's part of the GS expression systems kind of moved on to accommodate multi-chained formats with high expression. So we have that coupled with piggyback, so we are ensuring that we're getting kind of higher expression for molecules that would traditionally be more difficult to express, so they have trouble folding, and therefore that would usually manifest itself as kind of lower secreted titer. So by all these technologies that I've talked about previously, boosting titer, that all helps, and those are all available. They're all packaged into the GS expression system. Alongside kind of the vectorology and the host cell lines, you also have the production system that we use. So we mentioned there is the intensified platform, but we also have the TheraPro fed batch platform, and this is built upon kind of the previous production versions that Lonza's continuously improved on over the years. And so TheraPro simplifies all of that previous history into a much more easy to use production platform. So it's a single media and a single feed, and we get the same product expression and product quality that we've seen with the kind of more all singing, all dancing versions of the Lonza production platform. So the whole Lonza GS system is packaged together so that it's completely portable. We use the same methods and materials in-house as we license out to GS REA holders. So that allows customers can potentially take the same tools and technologies in-house to start their cell line development and then transfer it into Lonza, and it's made to be as seamless as possible so that we can take a cell line and drop it straight into our manufacturing facilities if need be. So it's completely portable. Yep.
So let's talk development. We've devised our early development strategy. We've selected a candidate, an optimal vector design and expression system that we know can take us all the way through the clinic and to the market. Alice, how do you assess whether that molecule fits within a platform or needs bespoke development? And how does that impact timelines?
So it's a really exciting time, I think, where molecules come to us for assessment for development and manufacturing. So we generally get all of the scientific and technical experts together along with our proposal teams and work through with the customer what their key requirements are in terms of the molecule, what their sort of quality attributes and timelines, things like that. And so then once we start the initial assessment, we look at how close it aligns to Lonza platform processes. And so, as Megan's discussed, we look at the exceed and the likelihood that the molecule will be successful in the exceed system. The vast majority of molecules are, as Megan's mentioned. But we also need to look at the purification process, and is it a standard purification process? So is there an FC portion on the molecule? Will it fit within a platform protein A procedure? And also in terms of the clearance of other specific product related impurities, and this can be much deeper than a standard monoclonal antibody, because quite often there are product specific impurities that are very challenging to clear. And so we have a toolbox approach to development of our downstream processes, so we can rapidly screen multiple resins, not just the polishing resins to remove things like homodimers from bispecific antibody molecules, but also we find with these new molecular formats, they're quite often sensitive to pH or sensitive to capture conditions. So we can tailor the whole purification approach, right from the first column, the first capture column, right the way through to the final polishing and every step in between. And because we have a toolbox approach and we've tried to think of all the different conditions that we could use, we can put them on a high throughput automated screening system. And so it doesn't actually impact our timeline significantly at all if we're working within that toolbox. Obviously, there are some times where even outside of our toolboxes, we have to develop things, and that can impact can impact timelines. But in general, our toolbox approach allows for minimal disruption to our timelines, because we're already anticipating the challenges that we might see with these more complex molecules. Similarly with analytics, so we have a wide range of platform analytics, and the advantage of using platform is you have a relatively fast workflow into the development program. You can get good indications early on. As we mentioned, we try and use the same types of analytics for the earliest development all the way through to QC. And we find that with our platform analytics, the vast majority of molecules will fit within that. So we've developed the methods, we validated the methods so that we know that a lot of molecules that we see will fit within that. So you don't have to do additional development or validation work. There are always exceptions to that rule though, and we will see molecules that don't fit within our platform analytics. So quite often because they're too large or too small, or sometimes they're too heterogeneous to fit within the methods that we've developed. In those cases, we use a similar approach to we would with our purification development, in that we've developed, and our R&D teams within Lonza have developed a wide range of toolbox methods. So for example, one of the sort of key analytical processes is to look at the titer of a molecule coming through the development. So how much is your cell producing? And we normally do that using a protein A capture. Not all molecules have protein A, and we use an instrument called an octet. So we've developed a whole suite of methods in which you can do other molecules that don't have an FC portion, other ways of capturing that, other ways of measuring the productivity. Similarly with bispecifics, one of the key requirements is that you don't have product related impurities and that you can quantify that. Those methods aren't platform necessarily because they're unique to the molecule. So we have a whole range of mass spectrometry toolbox methods that we can pull one of those off the shelf in order to determine if the molecule is correctly assembled, and it will fit to the molecule. So in general, if we're using those kind of toolbox approaches, we can fit those within our current timelines. They don't really add much to the timelines. However, if you're developing completely bespoke, this can add additional time to your timeline, but we work with really expert teams to minimize those risks and prioritize them, and work with the customer on the technical decisions and getting those risks or development challenges highlighted as early as we possibly can.
What about in preclinical studies? So how do you ensure a successful formulation for those preclinical studies?
Yeah, so with the formulation, it works quite similar actually to our analytics and our purification strategy, is that we have a toolbox of formulations that we can rely on. So we have a huge experience within Lonza of what formulations work and what formulations don't work, or what formulations are likely to work with the molecule. So we will select the correct steps based on the molecule type and the complexity of the molecule and our prior knowledge of those types of molecules. And so then from that point, we can use our experience for the selection of excipients and what the development steps that are needed are. And so we bring in the formulation study early on, and we have quite innovative high throughput screens for formulation. And then we put the molecules onto early formulation stability studies. So we'll have a panel of formulations that we can assess which formulation will work best for the molecule. So it's really based on our experience, our knowledge, what we know about those types of molecules, and the tools and the toolboxes of the formulations that we've developed within Lonza. We also have conversations with our customers. Sometimes they have a lot of knowledge of what formulations do and don't work for their particular molecule.
So we've covered the importance of these early development strategies, having the right analytical, downstream formulation process in place. You know, there's a lot of factors that a drug developer needs to consider as they're moving towards the clinic. But one that comes up time and again is how fast are we going to get there? Megan, given how critical timelines to the clinic are for biotechs, how do you help them just make things easier and navigate that journey?
So, as we've said several times through here, it really depends on what the customer's needs are, what their goals are, and what their molecule is. So we've got several kind of baseline approaches that we can start with as discussion points with the customer to show kind of what an example timeline would be. And then we just bespoke it depending on their risk profile and what they need to achieve. So when we talk about, so often a conversation will go, what is the fastest I can possibly go to IND for a given molecule? We will start with looking at, okay, is speed to tox critical in this particular project? If so, we've got several kind of accelerated enhanced tox programs that we can look at. And so what one of these might consist of is looking at if we can use a pooled approach to go to tox. So as we said before, we can get tox material in 2.5 months if we're using a pool and we've already got a formulation that we need to that we can go into. If we need to potentially look at formulation work, we can use pools, we can generate some early EDS material, as we mentioned before, to start screening formulations. So we can leverage techniques like design experiments and high throughput screening in order to reduce the time it takes to screen those formulations and the amount of material we need to. And this can actually knock off about 6 weeks off a formulation timeline. We can also look at establishing assays quite early on so that they're already ready when we have a clonal cell line, and then start producing pilot and first GMP batches. So that can be done off of kind of early material supply, again, off pools. And one of the reasons we can actually go down using these kind of pool approaches rather than clones is that we can leverage all the experience that we have with generating pooled material and then compare the product that we're making from pools to clones. And we're very confident the material that we get back out is representative and consistent. So we are confident we can draw those conclusions, again, based on the data that we've generated over many, many years. So all of these kind of different approaches, we can combine to really work with a customer and figure out what the fastest timeline we can build with them is to meet their requirements and their needs.
I guess if speed is something that everybody's also focused on, regulations are the other side of the coin there. How do these really early development decisions impact the biotech's ability to meet regulators later on in the process, get those requirements right, particularly when it comes to the complex biologics that Lonza is dealing with?
Yep. So, like I said before, the regulatory filings are always kind of in the back of our mind. So it's making sure we're documenting everything and meeting the requirements that are expected of various regulatory agencies across the world. So when we look at things like vectorology, we want to kind of bear in mind what is the impact of any changes we make going to be on genetic characterization down the line. So we talked about kind of removing any kind of sequence liabilities via the manufacturability assessment that we can do in silico quite early on. And then there's other options that we can build into a timeline, again, based on risk profile of a customer. So if there are concerns about sequence errors, so nature is not perfect, no matter how well we do things, sequence changes can be introduced just kind of naturally. We can screen the research cell banks quite early on, either looking at the mRNA or the protein sequence. These are options that we can build in so that there are no surprises later on. We can tick that box saying we know that the product that you're intending to manufacture is correct. We've kind of touched on clones versus pools. This is a little bit of a hot topic. So when can you use pools to really accelerate your approach, and will it meet the regulatory requirements? And so, as I said before, we continue to build that body of data showing that you get comparability between both the pools and the clones, right? So that right now, while we're using pools to do some tox studies, down the line, we may look at potentially using those pools maybe to support phase 1 studies. So this has already been demonstrated by a pandemic approach, so those really accelerated timelines. But again, as we continue to build the data sets, this may help accelerate timelines even further. And then another one that comes up a lot is cell line stability. So we always recommend that if a company wants to really de-risk their process, they can do a development cell line stability study where you're basically de-risking your LIVCA batch, so your in vitro cell age batch, much further down the line of commercialization by artificially aging those cells up and then assessing whether there is any loss of product expression, or from a regulatory point of view, you just want to make sure that there is no change in the product quality of what you're making. So we can offer that on critical path or off critical path, kind of at the development stage, so that you don't get surprises down the road. So again, it's all about balancing risk appetite, leveraging the historical experience we have, and then building solutions that fit customers' timelines and budgets.
Alice, a lot of the people who are listening to our podcast at Beyond Biotech, they're smaller biotech companies, they're out licensors, they're innovators, and a lot of them are also navigating that preclinical development path for the first time. With these new molecular formats, what sort of advice can you give to these biotechs and these founders to help them through that process?
I think, having worked with quite a number of these smaller companies now, especially those developing new molecular formats like bispecifics for the first time, I think a key piece of advice would be really don't underestimate the value of early and integrated thinking, and from the earliest possible moment start thinking about manufacturability, stability, and your analytical strategy right from the start. It can be really helpful for small companies to use CMC experts as soon as possible. Get someone on board who understands what the CMC risks are and what you might need to have a successful regulatory pathway. And these can really help you identify risks and avoid costly mistakes. For example, if your molecule is known to have aggregation or poor expression or charge heterogeneity, these can lead to increased costs and slower routes to clinic, and in some cases can really require costly rework. And getting the right partner on board, having a partner that really understands CMC and what these pitfalls might be, by avoiding them and focusing on optimal molecule design, can really help you to avoid these problems in the future. And it's important that small companies don't assume that your development partner can fix all of the problems associated with a bad molecule design. There are some things that can be fixed and overcome, but getting it right early on will certainly help a smoother path and a less costly path to the clinic. And having someone to guide you through that process is really invaluable. So it's important to get the right advice, but also to choose a partner that can grow with you. So small companies often need help with strategic and regulatory guidance, and being able to understand what the requirements are to scale their product past that first regulatory filing.
Yeah, getting those early decisions right, making the right choices early in development, that's always so important. And it sounds like Lonza is there to help. Megan, where can listeners learn more about Lonza? Where can we point them?
So I'm going to point you into the direction of our website, so Lonza.com. There's loads of information on there about the ranges of services that we offer and the various technologies that you can access both in-house and through our licensing team. And if you find the Contact Us button at the bottom of the website, that will help direct you to a variety of our global experts that would be more than happy to discuss any of the solutions we talked about today further with you.
Megan Mason, Alice Harrison, thanks for joining us on Beyond Biotech today.
Thanks for having us.
Thank you so much.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.